kind of a very, very scary term, right? But it comes from the physics. It's a kind of a system, robotic system, which explains the level of the chaos, the level of the entropy. The entropy is kind of order and disorder, yeah? More entropy you have, more disorder you will introduce to the system, and vice versa. So it's a kind of, when you have a mixture of, when you have a system, then, for instance, the computational fluid dynamic, last time we were talking about computational fluid dynamics, and in computational fluid dynamics, this particular concept is also working when you have a fluid shear stress kind of fluid dynamics, and then you mix it up, the fluid, and this mixture duly represents the ergodicity. It's a kind of system without the memory, some kind of averaging. When you have the averaging of the system, and you cannot really see the trajectory of the system like in the molecular dynamics, then you could say that the system becomes ergodic. So we try to implement this concept to this particular paradigm to come up with the mechanism of chromatopsis, because this is the kind of evaluation of the genomic chaos, because it becomes chaotic. You got a disintegration of chromosomes, everything is going to die, the cells, chromatrixes, chromatoplexes, chromorexes, the disintegration of the nuclei, yeah, and the plasma lemma will be also disintegrated, and that's what you see at the microscope. Okay, so, and this is kind of a project which is connected with the previous one, and we try to do some kind of protein target, let's say, protein, the target hunt, protein target hunt, to come up to some molecules, protein molecules in the body, where we can probably inhibit and also have some prognosis, better prognosis for the cancer patients. As you can see here, this is Professor Prof, and with a late stage patient. This is the very, very aggressive cancer patient you have here. It's called the Bellini sarcoma, carcinoma Bellini, duct carcinoma from the kidney, and usually the terminal stage, so you cannot really help the patient. So we're also trying to implement some kind of experimental approach, whether we can actually improve the survival rate of the patient. This is what exactly we are doing here. So this is the Bellini cancer excised from the human body, and we are, for this particular project, we are doing the sequencing of the cancer. We do RNA-seq, and from this, first of all, we are doing biopsy, and then of Bellini carcinoma, then we do the RNA-seq. From that, we trying to recreate protein-protein interaction networks, and hopefully we will come up with a protein-ligand complex where we can inhibit and reverse the cancer progression in a patient. And here's the result. I'm still trying to figure it out, the different parameters in RNA-seq, because RNA-seq will give you differential express genes. So you might generate different subsets of differential express genes, and you don't know what are the exact differential express genes which are probably giving you the best protein-protein PPI. Therefore, I'm using different statistical patterns, like different p-values, trying to figure it out, different subset of upregulated genes to find the better, so to say, stable generation of protein-protein interaction network. And so we got roughly 10,000 after the screening. We got roughly 10,000 genes. Then we converted the genes to, we decreased the number of the 10,000 genes to 300 genes. using the p-value and fold change parameters. It's well known in bioinformatics, of course. And we came up with a small number of genes, like 300, and then we were using, after that, we created, so to say, the representative network where combining 30 genes. So the idea was to investigate in how we can disintegrate this network, the disintegration network. And we are calling this strategy the network resilience. And we are trying to calculate some mathematical parameter, percolation coefficients, for instance. Percolation coefficient will give us the idea how many proteins we have to knock down here in the network in order to disintegrate. So the minimum amount of the networks in order to disintegrate. And I'm not going into detail, but of course we did all of this. Now the paper is under revision in high impact factor journals, and we're proposing that for this particular network we need, like, the percolation coefficient is about 15, so we got 15% of hub proteins, the proteins with the high connectivity levels here, nodes with a high connectivity, we are calling them hubs. And these hubs are extremely important to knock down the whole network. Therefore, we are thinking if we will knock down this particular hubs, we will disintegrate this network more efficiently, because if you don't know about the information about the hubs, you have to do like randomly trying to knock down 85%, which is not really practical. And here we have some structure. This is one of the rock. This is the CCR5 inhibitor, which is widely used in the HIV to treat the HIV. We use CCR5. This is a co-receptor, CCR2, CCR5, P120. And so here's as you can see, the CCR5 is the receptor, is the target, as one of the hubs where we're thinking when we try to disintegrate, if we're trying to create the protein ligand interaction complex, we might try to have a chance to disintegrate the entire network. So this is the idea. Of course, this is a little bit rudimentary and simplified, because you have to use different targets here, but of course this is very, very, very hard to do. We have patients and we got doctors and we have some schemes, which were approved by the government. It's sometimes not easy to prescribe the particular drug which will concern the particular protein, because we have to explain. This is a lot of regulations, and right now this is really for us the obstacle as well. But at least we are trying to find we're trying to rationalize the treatment, and right now we have I would say the outcome is good, because the 5-year survival for this particular patient is known to be 7 months, and the patient is now stable for 2 years. So we have some progression here. So I think in the future we might be more, let's say, less constrained with the regulations, and we will try to figure it out. We will come up with the stable protein PPI networks, and more drugs we have, so we might be even more successful. Okay, so this project was done in Germany, was published in